I'm Jamie Waese. I'm here to talk to you about building data visualization tools for large biological data sets. Now, I actually don't come from a biology background. I come from the world of computer animation and children's television, educational television, where I've spent the past 20 years trying to tell stories with pictures. Here's a little data visualization of my CV, and there's very few science terms here, no biology. So I've also, in addition to catching up with my programming skills and everything else in data visualization, I'm also learning biology right now too. But that makes sense because data visualization is a multidisciplinary field. In order to be successful at it, you have to be, in some ways, a jack of all trades. You need to be able to figure out what is the story in your data. You need to be able to identify key items. You need to know the field that your data is about. And let's begin with a little bit of a sort of preface of what is data visualization. Data visualization is the study of how to present your data visually and be able to pull out the images from what could be a lot of random noise. So we're all used to seeing tables of numbers, but it's very difficult to identify trends and extract meaning from, say, population data. It makes a lot more sense to use that data to draw pictures. One could spend an hour looking at this picture and see and make connections and draw inferences that you just wouldn't see with a table like this. So why is data visualization important? Many of you are probably familiar with the work of Edward Tufte. He's one of the godfathers of the data visualization field. He actually does this wonderful chapter in this book here where he explores the Space Shuttle Challenger explosion in 1986, which a lot of people might have heard has something to do with faulty O-rings at the time. Well, in fact, he suggests that the tragedy of the space shuttle was not really a fault of engineering. It wasn't an engineering tragedy. It was actually a data visualization tragedy. Because prior to the launch of the shuttle, the engineers who had actually built the booster rockets were well aware of the fact that O-rings do not perform well in cold weather. And in fact, prior to the launch, one of the engineers, Morton Theocol, the company that made the booster rockets, was actually slamming the table and saying, we should not allow them to launch today because look, we have all this data that says it's not safe to fly in cold weather. And even after the explosion, in trying to explain their logic of why they didn't want the shuttle to fly, they prepared this beautiful data visualization chart here, which is full of pretty pictures of rockets. But it's so hard to extract any meaning from this. So Edward Tufte actually took all that data and planted it in just sort of a very simple scatter plot. You can do this in Excel practically. And he plotted the outdoor temperature with a simple scale of damage to O-rings. And had the engineers actually presented the information like this prior to the launch, it would have been very unlikely that the flight commanders would have authorized it to take off. This is how cold, this was the outdoor temperature on the day of the launch. If you could just present your data in a nice and sort of simple and intuitive way, people might listen. You might see what you're trying to say. Okay, so how can we be more effective at data visualization? Like I said, it is a multidisciplinary field, and there's a couple researchers named Healy and Enns who are actually a multidisciplinary team themselves. Healy is a computer scientist at North Carolina, and Enns, J.T. Enns, is a cognitive psychologist over at UBC who wrote a paper recently that explores the notion of pre-attentive processing. And basically, the notion that we're very good at seeing certain things and recognizing certain sort of visual properties pre-attentively. We don't even have to think in order to see orientation changes, hue changes, length changes. Even in the time that I'm talking, I imagine a bunch of you were able to sort of pick out the difference in these images. But by the same token, while we can see certain things pretty attentively, the project, it becomes much more difficult to see things when you layer multiple sort of when you give people multiple visual problems. So if we try and, in these images, try and recognize the presence or absence of a horizontal blue rectangle, very, very, very hard, because first you have to go through and actually say, well, what's blue in this image? And then you have to go through and say, well, what's the orientation of these symbols? So I highlight them. But the more times, the more conjunctive searches you need to do, first I'm looking for this, then I'm looking for that, then I'm looking for this other thing, the harder it is for your audience to make connections and draw inferences from your data. Here's another one. These before and after pictures. What's different between these two images? As it happens, the reason that these sorts of games are fun is because we're blind to, it's called change blindness. I circle the differences. You can see, I mean, it's fun to play that game, but keep this in mind as I go through my next few slides, where I'm going to explore some data visualization paradigms in biology that many of you are probably used to. So we have bar charts with very complicated legends that, so if we want to sort of draw inferences and make connections between the values of these charts, we need to go back up and say, well, what does red correspond to? Well, here's tables of graphs that are color-coded. Track viewers. Talk about change blindness. Here we have to look at the clustering of information on different layers and compare, well, the presence or absence of lumps up here, how does that relate to the presence or absence of lumps down there? Here's more track viewers. Here's a track viewer that's wrapped in a circle, which looks very pretty, but suffers from similar problems. There's heat maps. I actually love heat maps. They're not necessarily pretty, but you can actually layer quite a lot of information into very little space. Here's a heat map that was rendered in a 3D game engine called Unity 3D. So it's a heat map where they brought the camera from the top looking down onto a 3/4 view, which allows you to sort of see differences in scale very nicely, but now you can only see a small portion of the heat map. So there's trade-offs there. We go networking diagrams and 3D models. 3D models are really cool, but there's all sorts of challenges in 3D ones as well. There's orientation problems. We don't necessarily, although we live in a 3D world, we don't necessarily think in a 3D way. So what are we even looking at? And this is such an abstract concept to begin with, and we map it out 3D and we put labels on it. It becomes very confusing to spin around. We have network diagrams, really complicated network diagrams. EFP browsers, I like this. Actually, Nick, I believe, developed the EFP browser, and is one of the reasons I'm doing a data visualization degree with Nick is because of the work he's done with EFP browsers. And some of you might even know the data visualization tools just put up here at the bar. Anyways, I'm in the process of updating some of these tools. So here's the Arabidopsis EFP browser that was published now 5 years ago. I'm in the process of taking what used to be allow you to type in one gene and see the expression pattern of that gene and where it's expressed in a plant, sort of using a heat map concept. Going from PNG into vectorized files, and also making it a lot more interactive. So the idea would be you can pick your plant, and you can enter up to 40 genes at a time. You press go. This is only 20, but it goes back to there. And then you can scroll up and down your list of genes, and actually you see a heat map there just sort of to compare expression patterns. And all of these things light up in real time. So that's one of the important things about data visualization is, can you actually present the data for your audience to interact with it and flip between pages and different views and change your thresholds and can you go from absolute to relative? So that's some of what this tool does. We have Arabidopsis, we have tomato, we have potato, as we move forward, we're starting to look at integrating different layers of data at the same time. So here's a little mock-up. I was talking about storyboarding earlier. Here's a little mock-up of what a tool might look like that allows you to look at RNA sequence data at the same time as EFP data. Here's a slide from Lima Sinha, where she has, it's an integrative data viewer, where she has phenotype data, or phenotype data up here, and she's connected it to regions of the chromosomes that are responsible for these phenotypes in two kinds of tomato plant. And there's actually quite a lot of data there that she's wrapped up neatly into a pretty little circle. It's not really labeled in a very clear way, but it's actually quite an impressive view. Now, for my thesis, I'm going to be working on a browser with Nick that allows us to explore data from the kilometer level all the way down to the micrometer and nanometer level, and then allow the user to be able to sort of slide between different plants and with little smart buttons that allow you to sort of apply, like look at this kind of view at the same time as that kind of view, and we're figuring that out right now. So, thank you for having me. I want to say thanks to Nick and everyone in his lab, especially Hardeep, who is right here, who wrote these very clever web service tools that power our EFP browsers. Thank you.